Oh, Global NMR discussion. I didn't see you there. Welcome to our lab. Well, if you're here, you might as well join us. I will tell you about a project we've been working on, how we probe dynamics in intact viruses like these.
So, our goal in this research was to establish a protocol that can be used to probe site-specific dynamics in large intact systems in a manner that is both simultaneous and automated. And we chose to demonstrate it on the intact FD bacteriophage major code protein as a model system. We did it by conducting pseudo three-dimensional experiments. We had carbon-carbon correlation with various mixing schemes, both J-based and dipolar-based, so we would be sensitive to both the rigid and the flexible regions of the protein. We also utilized REDOR-block as a mean to quantify the dynamics, since it allows measuring the motionally average dipolar interaction constant. And when compared to the rigid case, we get the order parameter, which relates to the amplitude of motion. Once we conducted all experiments, we loaded the results to Raven, a software developed in our group, and it is right here, free to download. And with all our scripts, we get automatically a list of all the dipoles, the carbon-nitrogen dipoles that can be detected. And here you see a plot of the backbone alpha carbon-nitrogen dipoles, order parameters. We have the entirety of the N-terminus. You can see that it's flexible, and then the order parameter increases gradually until we get something that is somewhat uniform. And that is interesting on its own because we can already see that it is flexible to some degree at millisecond timescale. But in previous reports, it was shown that the co-protein is actually rigid at the sub-millisecond timescales, meaning that we effectively narrowed down the window of the amplitude motion for large amplitude motions in the major code protein of the virus. In addition, we can probe dipoles of residues that contain CN bond. This is shown here. We have this lysine side chain that is quite flexible, but these residues, the lysines over here, are in the DNA binding domain, meaning that they are close to the negatively charged DNA, and that restricts their motion, and it is reflected in the elevated order parameter. So overall, we managed to have some sort of automated simultaneous protocol, and we are eager for you to try it out. So please, if you're interested to read more, please check out our manuscript. We recently uploaded to the bioRxiv, and thank you so much for listening.